Welcome to today's lecture on the process of protein degradation in cells. Proteins are fundamental molecules that perform various essential functions within cells. However, maintaining a balance between protein synthesis and degradation is crucial for cellular homeostasis. In this lecture, we will explore the intricate mechanisms involved in protein degradation, focusing on topics such as the Ubiquitin-Proteasome system, autophagy, protein quality control, protein degradation during aging, protein degradation in neurodegenerative diseases, UPS and cancer, lysosomal degradation, ER-associated degradation, ERAD, protein degradation in infectious diseases, protein degradation and cell signaling, and protein turnover in muscle homeostasis. Let's dive in and explore the fascinating world of protein degradation. The Ubiquitin-Proteasome system, UPS, is one of the major pathways responsible for selective protein degradation in cells. It involves the tagging of proteins with a small protein called ubiquitin, leading to their recognition and subsequent degradation by the proteasome. The process begins with the activation of ubiquitin by an enzyme cascade known as the E1, E2, and E3 enzymes. E3 ligases then transfer ubiquitin to target proteins, marking them for degradation by the proteasome. This selective degradation plays a crucial role in regulating protein levels, removing damaged or misfolded proteins, and controlling cell cycle progression. Autophagy is another essential cellular process involved in protein degradation. It operates as a bulk degradation pathway, capturing and engulfing cellular components, including proteins, organelles, and cytoplasm, within double membrane vesicles called autophagosomes. These autophagosomes then fuse with lysosomes, forming autolysosomes where the cargo is degraded by lysosomal hydrolysis. Autophagy is essential for maintaining cellular homeostasis, clearing damaged proteins and organelles, and providing nutrients during starvation conditions. The proteolytic machinery consists of various enzymes responsible for the breakdown of proteins. Along with the proteasome and lysosomal hydrolysis, other proteases such as calpains and caspases contribute to protein degradation. Calpains are calcium-dependent proteases involved in cellular processes like apoptosis and muscle protein degradation. Caspases, on the other hand, play a crucial role in programmed cell death or apoptosis. Cells employ robust quality control mechanisms to ensure the proper folding, assembly, and maturation of proteins. These mechanisms help identify misfolded or aberrant proteins and prevent their accumulation or potential toxicity. Chaperone proteins, such as heat shock proteins, HSPs, assist in protein folding and refolding. Additionally, molecular chaperones and the Ubiquitin-Proteasome system collaborate to degrade misfolded proteins. Aging is associated with a decline in protein quality control mechanisms, leading to the accumulation of damaged and misfolded proteins. Impaired protein degradation pathways, such as the UPS and autophagy, contribute to the age-related protein accumulation and cellular dysfunction observed in aging tissues. Understanding the mechanisms underlying protein degradation during aging can provide insights into potential interventions to promote healthy aging. Neurodegenerative diseases, such as Alzheimer's, Parkinson's, and Huntington's, are characterized by the accumulation of specific proteins within neurons. Dysfunctional protein degradation pathways are believed to contribute to the aggregation and toxicity of these disease-associated proteins. Impaired UPS and autophagy, as well as defects in lysosomal function, play a role in the pathogenesis of neurodegenerative disorders. The Ubiquitin-Proteasome system has significant implications in cancer development and progression. Dysregulated protein degradation can result in abnormal protein turnover, leading to the accumulation of oncogenic proteins and the dysregulation of signaling pathways. Targeting the UPS components has emerged as a potential therapeutic strategy for cancer treatment. Lysosomes are membrane-bound organelles containing a variety of hydrolyses responsible for degrading cellular waste, including proteins. Lysosomal degradation plays a crucial role in the turnover of long-lived proteins, cellular remodeling, and nutrient recycling. Dysfunctional lysosomal degradation has been associated with lysosomal storage diseases and age-related disorders. The endoplasmic reticulum, ER, is responsible for synthesizing and folding secretory proteins. However, misfolded or aberrant proteins can accumulate within the ER, leading to ER stress and dysfunction. ER-associated degradation, ERAD, is a process that selectively targets misfolded proteins in the ER for degradation by the proteasome. This pathway is essential for maintaining ER homeostasis and preventing the accumulation of toxic proteins. Many intracellular pathogens manipulate the host cell's protein degradation pathways to promote their survival and replication. Pathogens utilize various strategies to interfere with the UPS, autophagy, or lysosomal degradation systems, enabling them to evade immune responses and establish persistent infections. Understanding how pathogens modulate protein degradation can provide insights into developing targeted therapies. Protein degradation plays a crucial role in regulating cellular signaling pathways. The targeted degradation of specific proteins can activate or terminate signaling events, ultimately shaping cellular responses. Examples include the degradation of regulatory proteins involved in the cell cycle, transcription factors, and signaling receptors. Muscle cells undergo continuous protein turnover for growth, repair, and maintenance. Protein degradation is essential for removing damaged or misfolded proteins and eliminating cellular components during muscle remodeling. Dysregulated protein turnover can lead to muscle wasting and various muscle disorders. In conclusion, protein degradation is a complex and highly regulated process that maintains cellular homeostasis by removing unwanted or aberrant proteins. The Ubiquitin-Proteasome system, autophagy, and other proteolytic machinery are responsible for protein degradation in various cellular contexts. Dysfunctional protein degradation pathways can contribute to aging, neurodegenerative diseases, cancer, and infectious diseases. Understanding the mechanisms of protein degradation provides insights into potential therapeutic targets for various diseases. Thank you for joining today's lecture, and we hope you have gained a comprehensive understanding of protein degradation in cells.